Everyone seems to be thinking about it. Anyone who wants to share, what do you think? Sanchita, what do you think?
I don't think that computers can cure diseases.
They can't cure. I'm sure they can't replace doctors, but do you think they can help us in curing diseases?
I don't think so.
You don't think so? All right. Anyone else? Would you like to share? What do you think?
They
Yes,
track
once.
our health record.
Once you have your hand up.
Ma'am, I also don't think so. We can replace robot with doctor, but I think they can help doctors.
All right. All right. Arush, you also have your hand up. Anything you want to add?
Ma'am, like I don't think so. We can completely replace the doctors by the robots, but they can help most. It is like next to impossible to replace the robots to the doctor.
All right. All right. So everyone seems very skeptical about whether computers can help us in healthcare or not. So I'd like to now invite our scientist for today, which is Dr. Lipi Thukral. You can all see her on the screen. I'll also just spotlight so everyone can see her better. Yeah. And she's going to tell us more about what we've discussed, that how can computers help us in curing diseases. That's what we're going to talk about today. So without further ado, I'm just going to hand it over to you, Dr. Lipi.
Thank you, Sharanya. And hello, everyone. I am Lipi Thukral, and I'm a scientist at an institute which is in Delhi. Its name is Institute of Genomics and Integrative Biology. And I'm really happy to be here speaking with you all about what kind of research we do in the lab. And also what I largely will try to convey you all is that there are different fields in computers and biology that one can do and play around with a lot. So I like to share my screen. Can I share my screen? And if you can help me out to say that you can see it.
Yes, ma'am, it's visible.
Yeah, okay. Okay.
Yes, ma'am.
Thank you. So whenever I get lost, just try to tell me where, I am not able to, probably you're not able to understand anything, or you need certain other suggestions to help better. I can elaborate on that point. So please help me during my presentation. And what I'm going to show you, I think everyone has seen a computer. There isn't anyone, because we are communicating via computer. So we have an idea about what computers can do. But there's a very, very interesting thing which I would like to bring to your attention, that biology, which is a different stream, which you might now study in detail in coming of your classes, can be combined with the computers to give rise to a new field, which is called as computational biology. And this field is not really new. It has been here now for around 20 years, but relatively new. What do I mean by computational biology? I definitely don't mean just the addition of these two. I mean that computer applications can be used in biology. And that's a very fascinating way to understand what happens in our body. Okay, so one way which most of you replied to Sharanya's answers were, how robots can replace the doctors. But the other idea is also not medicine, but biology research. Like in biology, we study about human body. And we can, in fact, in my lab, we use computers to study about human body. You all know about human body, don't you? Do you know about human body? We have brain, we have so many specific organs that perform function, but there are very interesting molecules in our body, which I'm going to talk about, which we study. So this start of the presentation is also to give you an idea that different fields can be merged. Like, for example, you go to the school and have different periods of different classes. Like you study English separately, you study Hindi separately, you study science separately. But later, when we do work, we can also combine these subjects. For example, biology can be combined with chemistry. You can also now combine history and mathematics. So now people love to put two things together because they give you a very nice view. Let's say you're seeing a tree from top of the building, you see a different tree. But when you see a tree from down below, you see a different tree. So the idea of merging the field is very interesting because you gain different perspectives. So in bioinformatics, for example,
Because come
2 to 5.
so
2 to 5.
in bioinformatics, for example, you can apply chemistry, biochemistry, biology, statistics, and also mathematics. And let's see what we do. We use computers. The question you can ask me is that what is the need to use computers, right? So why do we need to, we are studying human bodies, we can do in laboratories, analyze what our hands like and so on and so forth. But what has now happened, and you'll be surprised to know that all of the techniques that happens in the lab, which scientists produce, is a lot of data. So inside the wet lab, which is called as the experimental methods of biology, we produce a lot of data. And all of you would know that Google, for example, is producing a lot of data. When we click their ads, a lot of information goes to their servers. So you can see there's a lot of data. And similarly, if you can know, there are also these handwear devices. Do you know that it can collect the blood pressure? And also there are devices that can collect your pulse through watch. Do you know of such a watch? So there are watch which can now detect a lot of your movements and biological data. And now imagine, this is so much of data in 24 hours, where how you sleep is a data, how you get up is a data. And similarly, in experiments also, we have a lot of data, but how do we understand that data? So what we do is that in order to understand the meaning of the data, we write computer programs. Do you know what are computer programs? There are computer programs which can help us understand the meaning or try to process the data. Okay, and this is what we do, computer programming. Okay, so let me help you. Let's say if you go to a hospital and if you have to get an x-ray, which is not a nice thing, but let's say you've hurt your bone, and if you have to go and do an x-ray, there are now computer programs which can detect and say which kind of x-ray is healthy x-ray and which kind of x-ray is a diseased x-ray. So there are a lot of applications where one can find how computer programs can be used. So moving on, I want to show you the lab where I work. This is a very beautiful campus we have in Delhi. And if any one of you want to visit, just write to me. We can also arrange a visit and you can see how scientists work there. And we work on very nice experiments and then trying to spend a lot of time, which is not nice, but we have to do a lot of research in trying to understand questions which no one has solved, right? So this is the place where we all work. And my lab works on a theme which is called computational biology. We try to mimic what is going on inside the cell. So let's say this particular condition, can you see this is a duck and a cat is sitting on it? In the same way, in our body, we have sometimes proteins and different DNA molecules which sit with each other. And we want to understand how they're sitting, and we can then simulate that. So I like to show you a lot of movies. I will show you to make you understand. So for example, This is a roller coaster. This is a child.
Ma'am, I think it's a VR.
Yes, it's not a VR, but it's trying to mimic a VR by a parent. So now the parent, see,
Ma'am, it is just to entertain the child,
sorry,
like in something real, which is perfectly 3D,
yes,
or which makes it real.
yes, yes. So it is trying, the parent is trying to make the small child understand that probably she's sitting on the roller coaster by trying to mimic the whole situation. So these are called simulations. We are not in the same environment, but we are trying to give the reception that you are undergoing a roller coaster. So similarly, what happens inside the cell, we try to recreate in a computer. Now, let me show you examples of those movies. In our body, we have a lot of molecules, which I will come to, but they all move. Imagine you have these proteins which are moving like the lens aperture, going big and small. And you have the skin. The skin is made up of lipids, membranes. And imagine you don't see them. They are very small, but they're all moving. These are the lipids of a membrane, which is there on the skin. And why do they move? I'm going to talk so much about that as well, but these movies will firstly orient you in trying to understand. This is a movie by my research student, uh, Surbhi, who has been working on this protein for 5 years, and she's come up with very nice understanding of how this protein works. Look, this protein, how it moved and how it comes together. This helps in communication between the cells. And when we know that it comes closer and how it performs functions. So this is a very nice work she did. And then later, this is a membrane which you are seeing in the white, and this is a protein which is coming close to the membrane. Look, this movie. Now protein is going to come and sit. It's sitting now, and it has got attached to the protein. These all terms I'm going to now elaborate more, but one more term before I go into fundamental proteins and more things to talk about. You all know that we are still in a pandemic, although it has got into a much more relaxed stage. We all know now our SARS-CoV-2 virus, do we not? We all know. So this is that virus, and it's a depiction by one of an animation person who's made a virus sketch. And this sketch is showing a protein which is sitting outside the virus. It's called a spike protein. Okay, what is it called? A spike protein. So we did a lot of work around the spike protein. See, this is the virus. And it had a lot of spike protein, which helped it to attach to our throat. And that is why we have these flu-like conditions, because it has infected our throat. I'll show you this movie again. Look, this is the entire virus. Inside it is a DNA or RNA. You can have both in the virus, but in SARS-CoV-2, we have RNA inside it. And then outside the virus, we have the protein molecules. Okay. Now you'll have a lot of questions about why proteins, what are DNA, but just last thing to show you again a movie, that these proteins bind to proteins to perform a function. Gray is another protein and red is another protein. And this might be not with very zoomed resolution, but they're both different proteins which are binding to the same protein. So it's a lot of questions that we can address.
I understood one thing.
Yes, please tell me.
The virus proteins stick into our throat and then release the virus RNA in our throat.
Yeah,
which we get infected.
Yes, that's excellent, Advik. And this is very nice that you could say that because what releases and attaches is that are those spike protein. So can I tell you what is a protein? Can I tell you what is a protein? All right. So we have a lot of molecules. There are DNA. DNA is one kind of a molecule. There is protein and there is also lipids. So these are all the molecules inside a body. Let me tell you, in your house, you have a big kitchen and you have very small utensils. And these all utensils, they function to form something. So you can imagine these small molecules to be residing inside your kitchen, which can be a mitochondria, which can be a larger nucleus. So these are large houses in the cell, but small structures of your body are these molecules. And these molecules are known as DNA, protein, and lipids. DNA looks like this. Protein is something which I work on. So I'm going to talk to you about proteins. Okay. And I'm going to talk to you also about how protein goes wrong in a disease and how drugs and computational biology have been very important in this direction. So I'm going to now give you an idea what is a protein. Actually, a protein is made up of even smaller molecules, which are called as amino acids. So they are like beads. Like, you know, we have mala and we have these very small beads which can come together, but small beads by itself are not proteins. When amino acids form in a linear combination and of different shapes, they form these proteins. So you see, they are all separate, then they come together and look, they have formed a shape. That's very fascinating. Because that's another point too, Advik, to remember that proteins form different shapes. And to all of you, I'm trying to tell you this, that protein is not one shape, it's of very different shapes. So I can see some of your screens, you have very nice shapes. I can see Johanna's shapes. You have a very nice wall where you have a pink flower and you have a banana. So these are different shapes. So this could be also protein shapes, which are performing different functions, okay? And these functions are arranged in a lot of different hierarchy. We can look at here, look, this protein is rather large. It has come from this amino acid sequence to form something which we call as secondary structure, then even bigger protein, it's called tertiary structure, and even further beyond is called quaternary structure. So they all come together. So for example, the entire flowers come together to form bouquet, and that's where we have very interesting quaternary protein structure. There's a hand up. Vansh, do you want to go ahead and speak?
Yes, ma'am.
Please ask.
Ma'am, are they like the consumers, primary consumer, secondary, tertiary consumer, quaternary consumers?
They're not consumers, but they're building blocks. So we all have played, and you all have played with Lego blocks. You know Lego blocks? So when you have individual Lego blocks, that's the primary chain. But when you make a building out of the Lego, that's the protein. So a protein is formed from these small blocks. And then this larger tower is called the protein. So you can see Lego is not a consumer, but a building block. Does that help? Have you played with Lego?
Lego is primary, and then it changes into secondary, then tertiary, and then quaternary.
Yeah, you know, wow, that's amazing. This is what I was trying to convey. And thank you. This has very, very encouraging that you're understanding. And I'll go further and also tell me.
And I'm also taking notes in the chat.
You're taking notes. Wow. Okay. I'll be extra particular about it. And I will now talk to you about different kind of proteins. And I'm going to talk to you something about scary thing. Is it okay? Are you scared easily? Are all of you scared easily?
No.
Right? Exactly. I thought that. I imagined it because I thought that you all are anyways attending science lectures, so you could not be scared. But I'm going to tell you about a protein which is toxin, snake venom. So for example, when snake bites, it releases a protein which is a toxin. And when it enters our body, it could really harm us. So there are now drugs that are available that inactivate toxins. But again, what it releases, you can see is as a liquid, but inside the liquid is actually proteins. Okay, these are called toxins. I have again a hand up by Aisha.
Yeah, I was just asking, so like all proteins are not like helpful. These are all of them are not healthy or like good.
Yeah. So I'm going to talk about that very soon, that what happens when a protein is not healthy. So can you give me some moments and then I come to that? Okay, thank you. So yeah, I'm going to talk about that as well, but I'm right now on this slide, you can see different kind of protein names. You can also see that some that like to store, some that have hormones like insulin. If some of you know patients which are diabetic, has a very strong relation with a protein called insulin. I will show you that as well. There are some, blood, like we all know blood, right? It's red in color, but it's actually a protein called hemoglobin. What is the name? This is the name, hemoglobin. Okay. You have, Arush, you want to ask something?
Hemoglobin contains like oxygen, mom. It carries oxygen.
Yeah. I mean, I'm so happy that you know it already. It carries oxygen, but you know how? Because it has the structure which I'm talking about, and oxygen comes and binds at 4 places. So it's very specific molecule. It's a protein molecule that binds to oxygen and takes the oxygen everywhere. Because now if I am, let's say, running a lot and I need more oxygen,
Can
then
you show the molecule?
yeah, okay, you want to see the molecule? I showed to so many proteins. So this is the protein, and this would be something like oxygen, the red color, a single, because oxygen is nothing but a complete O2, right? So you have not a large.
मतलब ये पूरा का पूरा ऐसे यहाँ से कैप्चर होता है और यहाँ पे स्टोर होता है.
हाँ बिल्कुल, इसके साथ बाइंड करता है क्योंकि ऑक्सीजन छोटा है, हीमोग्लोबिन बहुत बड़ा है, तो हीमोग्लोबिन सारे ऑक्सीजन को बाइंड करके पूरी बॉडी में सर्कुलेट करता है ताकि हमारी बॉडी में अच्छे से हर ऑर्गन को ऑक्सीजन मिल जाए. ओके, आई हैव अनदर हैंड्स अप फ्रॉम someone titled Dr. Smita. I have one more doctor in the room. Yes, please.
Ma'am, oxygen carries blood and it transported to every body part. No, ma'am, is there any body part which does not get oxygen from the blood and gets directly from the air?
The oxygen is directly reached to the lungs. How nice of you to ask. And oxygen is a way in which our lungs, the respiratory function of the lungs is respiration. No, we take in the oxygen and we take out the CO2. So that is the performance of the respiratory organs that are lungs. Okay, the rest of them are then distributed by the blood. Okay, can I move on? We have one more hands up, Arush.
What gives the blood the red color?
Wow, I have to say this, that I'm not prepared for these questions. I'm trying to think. Okay, so the color of the blood is actually from the protein, but how is it?
Mom, I think is it due to red blood cells and hemoglobin?
Yes. Yes. So the red blood cells have hemoglobin, which is a characteristic of the red color. You're right. You're absolutely right. And we also have white blood cells.
Yes, ma'am.
Yes, ma'am. Yeah.
And what do white blood cells do?
Ma'am, I'm not sure about those.
So white blood cells increase in our body when we have any kinds of infection. So you see, whenever
White blood. Ma'am, I also know there's a whole chapter about it.
yes, yes. In fact, there are people who are working years on it, on studying infection, because we couldn't understand how virus completely invades our body, infects us, and leaves us in a very tired and disease condition. So that is why we do research on how viruses attack the body. So I like to move on now. And I like to show you some examples of, again, how proteins, for example, help in different other kind of functions. So for example, if we have muscles contraction and relaxation, do you know the muscles can contract and relax? And this is done by very large proteins that are called as actin and myosin. We also have a lot of proteins in our intestine. They help us in our digestion systems. Okay, like for example, lipases, proteases, amylases, they all help to churn the protein that we eat, break it into smaller parts, and lets it absorb the body. Now, something we have already talked about a lot is how oxygen travels in the body, which is by hemoglobin. So all in all, proteins carry very important functions in our body. And as you can, I just want you to pay attention here. This was hemoglobin, this was oxygen. So now what happens is that I'm trying to make a concept, I'm trying to introduce a concept to you, which is the protein have a binding site and a ligand. So ligand was the oxygen molecule and protein is hemoglobin. They both bind in a very specific way, like a machine. Okay, and they have to be bound, like it cannot be like any other binding, it has to be at the same place. And when this happens, then we have the function. Okay, so there are a lot of ways in which it can go wrong.
अगर वो टेढ़ा टेढ़ा जाएगा तो वो बाउंड नहीं हो पाएगा,
वो बाउंड
फिर
भी
वो नहीं होगा,
नहीं होगा,
फिर वो सीधा का सीधा यहाँ फ्लोट करता रहेगा,
बिल्कुल,
मतलब उसे एक पर्टिकुलर वे में जाना पड़ेगा.
yes, and it has to be specific. आपको तो समझ भी आ गया, मैं तब से बोल रही थी स्पेसिफिक क्या होता है. लेकिन आपको लगता है कि जब वो स्पेसिफिक बाइंडिंग नहीं होगी, वो टेढ़ा जाएगा और नहीं अच्छे से बाइंड करेगा, तो फिर तो हमारी बॉडी में डिजीजेस हो सकते हैं, राइट? क्योंकि ऑक्सीजन नहीं मिलेगा हीमोग्लोबिन को, तो फिर so that's
अगर ये ज्यादा हो जाएगा तो फिर डिसीजेस हो सकते हैं.
Exactly. That is why that brings the whole point to the section 2 of this talk, that the proteins, when they are not healthy, that gives rise to diseases. Okay. And let's try to understand that through one more thing, which I want to say, that proteins can be active and inactive. Okay. Which means that, you know, when you all sleep, can you listen to a talk? You cannot, because you're not functional. In same way,
Ma'am, I know about pancreas.
You know about pancreas? Can you tell me about pancreas?
मैम मतलब ये पैंक्रियास इंसुलिन का लेवल, मतलब ये ही इंडिकेट करती है कि आपका ब्लड शुगर लेवल ज्यादा होगा तो फिर शुगर को वो स्टोर करा लेगी, और कम होगा तो फिर वापस बाहर निकाल देगी.
बट वो क्या, कौन सा प्रोटीन है जो शुगर के साथ बाइंड करता है? पता है उसका नाम क्या है? इंसुलिन.
हाँ, वो अभी मैं लिखा हूँ
ना, तो सबको भी मैं बता देती हूँ कि उसका नाम इंसुलिन है. And I'm now coming to Aisha's question, which you're raising the hand. Yeah, Aisha, you want to ask again?
Yeah, I had a different question this time. I was asking, isn't insulin a type of injection?
Yeah, right. So injection, yes, but what we are injecting is essentially a protein. So
okay,
that's how proteins are also coming in our body, which are helping us.
मेरे पास एक क्वेश्चन है.
हाँ जी, बताओ.
कि ये प्रोटीन का हम लोग लिक्विड कैसे बना लेते हैं?
प्रोटीन का हम लिक्विड कैसे बना लेते हैं? प्रोटीन्स जब हम लैब में काम करते हैं तो हम इतने इतने कल्चर के साथ काम करते हैं. प्रोटीन धीरे धीरे ऐड करके वो बड़े से कल्चर में बन जाता है. जैसे कि पानी भी है और हमारी बर्फ भी है, लेकिन वो कैसे चेंज हुआ? कंडीशन से चेंज हुआ. जब हमने ठंडा रखा,
मैं समझ गया, मतलब पहले पहले ये मॉलिक्यूल्स दूर दूर रहते हैं, मतलब बहुत सारे मॉलिक्यूल्स हैं ये दूर दूर रहते हैं और धीरे धीरे ये पास पास आके लिक्विड स्टेट में आ जाते हैं।
या एग्जैक्टली,
मैंने पढ़ा हुआ है।
और इसमें हम बफर्स भी डालते हैं, कुछ सॉल्यूशन डालते हैं, कुछ ऐसे केमिकल डालते हैं जिसमें ये बहुत
जिससे ये टूटे भी ना,
जिससे टूटे भी ना। एग्जैक्टली।
So then inside the insulin injection, there's insulin liquid which is injected into your body when you like inject, like from the injection.
Right. That's where we have these proteins. So that's how we convert the entire understanding of our protein into helping cure a disease. Okay. So now I think we've talked about this slide enough, that we have active and inactive form. But I want to now come to Aisha's question even further and trying to elaborate on the point that there can be a healthy state of a protein and there can be a disease state. There's hands up by Arush.
Insulin is mainly used like we intake insulin.
तो फिर तो ठीक है।
Mom, we intake insulin injections, mom, when we have cancer, mom.
No, not particularly cancer, but diabetes. Have you heard about diabetes?
Yes.
That's related to sugar level. So let me give you an example. Insulin is functional form is monomeric structure is like this. But when the insulin is not able to digest the glucose and it forms then bigger structures, then we have a problem which is called the diabetes.
नहीं मैं मतलब मेरे पास इतने शुगर है और इंसुलिन अगर काम कर गया
तो आपका कर रहा है,
इतना ज्यादा शुगर इधर वाला शुगर निकल जाएगा।
लेकिन बहुत ज्यादा शुगर नहीं खानी चाहिए।
पर अगर इतना ज्यादा शुगर होगा ना तो फिर इंसुलिन थोड़ा ही निकाल पाएगा इसमें से।
करेक्ट। सो देयरफोर
और हमारा शुगर लेवल बढ़ जाएगा।
Therefore the body,
पैनक्रियास कम रहेगा ना।
Therefore everyone has to have sugars in very control. And if you don't control your sugars, if you eat a lot of sugars, that's why parents say, no, don't eat too much of sugars because the body cannot digest the sugars. Okay. So we all will have limited sugars. Sugars is not something good for the body, but okay when we take in limited amounts. Can we move on to Huntington's disease? So this disease is something, a very bad condition and a disease when you cannot have any control on your body. Look, for example, the patients which have Huntington's disease have no control on their body movements. तो वो क्यों होता है? That is when this protein, HTT protein, has a lot of these red repeats. These reds are nothing but more number of amino acids which have come in the protein. So you can see when the protein was in healthy condition, Aisha, then it was able to shape in its required shape, which was performing the normal function. But when there are more repeats in the protein, something going wrong with the protein, let's say in its shape. Now the shape you can see, all of you, can you make out between two shapes? There are differences. Can you make out? This shape is abnormal shape, meaning it is not functional. And this non-functional state of the protein now leads to a lot of problems in locomotion, meaning when you cannot, yeah, Vansh.
Ma'am, is that dark red thing more sugars?
No, it's more amino acid. Again, the Lego, the amino acids. So there are more amino acids. Yeah. This, you'd asked me this previously, so you would understand that there are more amino acids, the Legos, and they had to be just 5 Legos, but now someone inserted 15 Legos. So obviously the building is going to come down because the foundation was not done properly. Aisha, you have another question?
Yeah. So is amino acids good or is it bad?
Sorry, I couldn't.
Is amino acid good for us or like?
For sure, amino acids are the building blocks of proteins. So, for example, there are certain very good amino acids which help us form more proteins, and there's some which are not so good amino acids as well.
मतलब मैं ऐसा बोल रहा हूँ
Can I answer Aisha's question and then I come to you? I'm
मैं
still
उसका एक्सप्लेनेशन ही दे रहा हूँ.
अच्छा, okay, okay.
मतलब आप अपने बिल्डिंग ब्लॉक फाउंडेशन में ज्यादा लगाओ. मैं ये बोल रहा हूँ.
नहीं, लेकिन अगर
बिल्डिंग
ज्यादा
ब्लॉक
लाओगे,
फाउंडेशन में ज्यादा लगाओ, आपकी बिल्डिंग नहीं गिरेगी.
but it has to be the right amount, Advik. For example, यहाँ पे ज्यादा
अगर ज्यादा होगी तो फिर अनबैलेंस हो जाएगी ना.
exactly. Now you have captured the point. So coming back to you, Aisha, when we the amino acids in its right state, the correct amount are really good for us. Okay, and anything which is leading to dysfunctional amount of amino acids, here we have more. The shape of the protein is changing, or something in the protein is changing. So then it's not appropriate. Okay.
So if you take too much of amino acids, it's gonna like you get this abnormal function, but then if you take the right amount, your body is gonna work fine, right?
Yes. Okay. So now we move on to other diseases. And there are a lot of diseases which I have put here. They are related to how proteins obtain shape. Have you heard about Alzheimer? Okay, Alzheimer disease mein we don't remember anything. So there are diseases which in the later stage of your life, some of the people who are struggling with Alzheimer cannot remember anything. And you know what has happened there? You have proteins which are simply have to be small. They have formed very large fibrils in the brain. So obviously, when you have very large structures in the brain, your brain is unable to remember. So it has a huge problem.
मतलब इसके अंदर मिसफोल्डेड प्रोटीन मतलब इससे हो सकता है डिसीज.
Yes.
इससे हो सकता है पर इससे नहीं हो सकता.
Yes, misfolded proteins are the root cause.
अबे अल्जाइमर्स मतलब हर किसी के पास ऐसा, जैसे कि सिकल एनीमिया का एग्जांपल ले लेता हूँ. ये फ्री प्रोटीन जो है वो है हीमोग्लोबिन. और ये ऐसे ही घूमते जा रहे हैं, क्या पता एक साथ कहीं जाके क्लॉट कर दे.
और ये क्लॉटिंग डिजीज की वजह से ही वी आर नॉट एबल टू हैव प्रॉपर मेमोरी और प्रॉपर ब्रेन फंक्शनिंग. ठीक है, तो स्पेशली सिकल सेल एनीमिया, one particular amino acid mutates. What does it do? It mutates. And mutates means amino acid gets changed from A to G, for example. So amino acids are also letters. And when they change, then you have diseases. I'm not going into that much detail, but if you have more questions, any of you, then please write to Sharanya and she's going to get back to me and I'll try to answer your questions. Okay. So now imagine what is going on is this, that what now you all have understood also, that when the protein function, there is an imbalance, it causes diseases. Okay. And those diseases can make you cough, can give you blood sugar problems, can also not make you remember. And there is a healthy state of proteins in which all of the proteins are functioning in a proper way. Okay. So how can medicine treat the diseases? And most of you have asked me around as well that what was insulin? You had so many questions. I'm going to give you example that when you fall and when you have an injury, what happens?
Ma'am,
yeah,
it's like if there is no prox, we are like blocking this way.
Advik, can you please use the chat to talk, please? Let ma'am finish. And if you have any questions, we can take it towards the end, please.
But Advik had a good point. So what happens is this, that there is a protein called COX-2. It releases prostaglandins, which cause inflammation and hurts us during the injury. When you take the medicine after the injury, which is ibuprofen, because you want the pain to stop. So this molecule called ibuprofen binds to COX-2, and therefore prostaglandins are not released in the body, and hence there is no inflammation. So the way medicines work, and now that you all know what are proteins, and you know, all of you know that they bind, right? Can you all give me a hands up that they all bind? So you can make drugs bind to that. And that is what, you know, drug discovery people do. They find molecules which are in the exact shape that helps the protein bind. So ibuprofen binds with COX-2, which is not leading to the formation of prostaglandins, and hence you have no pain. So you see, drugs are helping us to cure the diseases. Okay. And now, how do they bind? There's a lot of competition between what binds to what inside our body. You have normal binding, but if you give a lot of liquid, for example, you can have competitive inhibition and non-competitive inhibition of the binding. So you have to be very particular about understanding how the protein shape is to inhibit it. No, these drugs are called inhibitors because they inhibit the protein function. So what are the protein ways in which the experimental structures are there? There is an X-ray crystallography. There is also cryo-electron microscopy. There's also NMR spectroscopy. These are all the tools that help in understanding and seeing the shape, because wo itna chota hota hai. It's at angstrom level. Your body is in meters. Your cell are in micrometer, but proteins are even inside the cell, which means they are in nanometer. So you need to use these very fancy machines to see the proteins. And I have written all the machines through which you can see the proteins. And then we can also see the proteins on computers, which me and my lab works on. So you all, one of you said it, that show me the protein. So this is the protein. Can you see? This is the protein shape. And these are the secondary structure of the protein, alpha helix and beta sheet. And there's another way to see the protein here. And towards this, something is bound, which is, again, you've understood that they're inhibitors. So computational drug discovery is a field which allows us to discover inhibitors. And that will allow the protein to bind and stop it. So now it goes back to the first question which Sharanya was trying to ask, that do you think computers can help cure diseases? And this is how, if we know what is our target, what is the protein, we try to develop an inhibitor and stop the functioning of it when it is in a disease condition. Okay. And there is a very complex pipeline to develop the diseases, to develop the inhibition. You try to find out firstly, what is that protein which is helping us? And then you try to open in the computer, we try to obtain more information on the protein. You see a lot of molecules and you put a lot of ligands which can inhibit it. It doesn't work with one. You need thousands and thousands of screening to obtain the right shape. And then you shortlist these molecules, and then you again go back to the experiments. So you do a lot of it is in computers, but then you go back to the experiments and trying to validate it and trying to understand, okay, have you found the protein and have you found the inhibitor which has stopped the protein's function? Okay. So these are the drugs which I'm trying to say. Now, once you have the drug, it is working, then you can really do a lot of things. I will also try to tell you one more field, and I want you to look it up. There are a lot of proteins which have no shape because experimentalists have not been able to compute its shape or to experimentally determine its shape. That is where a very new discovery, AlphaFold, has come. Okay, AlphaFold has predicted all the structures, and it has come by a company which is called Google DeepMind. What is the name of the company? Google DeepMind. So Google has opened a company, DeepMind, in London, and it has put a lot of people together there which try to write programs So you see, now they have solved all the structures, or rather predicted all the structures which were not known before. Okay, so I hope you will now look up to AlphaFold. What is AlphaFold? What does the company do? I'm coming to my last segment of the presentation. In this is my favorite topic. How do antibiotics work? Antibiotics are also small molecules, and you would have all taken antibiotics if your infection is not going away. If the bacteria is accumulating in your body, then you take antibiotics. But there are molecules like penicillin, which is a form of antibiotic. An antibiotic then kills the bacteria so that you don't have the infection anymore. But you know, at the molecular level, this is how the antibiotic looks. And this large thing, I want to now write it somehow. I want to use a highlighter. This entire thing is the bacterial membrane. Look how thick it is. It is not even like our membrane. So bacterial membranes are really thick. To attack it, we use these molecules which are called antibiotics. And then antibiotic comes, invades the bacterial membrane, and then kills the bacteria. Okay, it kills the bacteria, because you know that if something goes very deep under your skin, you will completely disrupt that. No, a lot of blood will come out. So similarly, bacterial cells are killed by antibiotic molecules. And this is, I think, a very interesting lesson for you to know that membranes, which is the first site, is very important. And I want to stop the presentation here by showing you a membrane cake. This is the cake which I made, and this has a lot of lipids. I didn't have the time to show you what kind of structure it is, but the lipids are made up of these large phosphate oxygen molecules with large carbon chain, and you have proteins sitting all over the membrane. So maybe some other time I can elaborate on those membranes, but yeah, everyone had this cake. So this was very nice. So you would know that they're fluid in nature, membranes. They have these very nice chemical character, which is hydrophobic in nature from inside and hydrophilic in nature from outside. And that's how membranes protects us. They protect all our cells, they protect all our body. And this is hopefully the end. And I'm open to questions now, Sharanya.
That looks like a very delicious membrane. Thank you so much for your time, Dr. Lipi. We do still have some 5-7 minutes left. So I can see a few children have their hands up. So we'll go one by one. We'll start with Vansh. Vansh, do you have a question?
Yes, ma'am. Ma'am, but some viruses are too big that they cannot be easily done by the, that medicine, removed by the medicine.
In fact, you will be very surprised to know, Vansh, that we do not have very good medicines for viruses. We don't. And that is why this pandemic took so long. What we have are vaccines. Okay. And vaccines and drugs are two different things. Vaccines help prepare our body towards infection. So what vaccines do is that they elicit a response in our body such that the next time viruses come, we can have a normal response. But what you're asking is a drug. And a drug is very difficult against viruses because of what you said. They're either too large and we cannot know how to attack them, because when we attack them, they're also inside our cells. So we do not know how to distinguish the virus with our body. There are several drugs, but it's very difficult to have a very nice drug which is specific for a virus. We have vaccines.
Okay, ma'am.
Right. I can see Sanjay also has a question. Sanjay.
Ma'am,
Antivirals work.
Yes, they do work, but they're very less. As I said, they're very challenging. Antiviral is a term for everyone. I want to say Sanjay knows about antiviral, and I congratulate you for reading about them. The antiviral is again the drugs which are specific to viruses. So, for example, antibiotics are for bacteria, and antivirals are for viruses. They are there. We have a lot of antiviral molecules. And recently also for SARS-CoV-2, there has been a molecule which is proposed to be an antiviral. I'm saying it's not that effective. That's all.
Okay.
Krishika, you want to go next?
Actually, when did this virus and stuff come? Originated?
I cannot hear her.
I think what Krishika is trying to ask is where, or say when did viruses originate, like in the world?
Right. Okay, so Krishika, this is a very interesting question. This is almost like saying that when did we all come? But there were very small microorganisms when everyone started to evolve, including human life. So, you know, we have all evolved from gorillas. You all know this, that in many several years back, human body was not even as we see now. We had apes as our ancestors. So even going back to that age, they were very small archaeobacteria. They're very small, and even then we had viruses and bacteria. So a very small cell, a single cell like amoeba, evolved to form multiple cells, and that's where the evolution of other microorganisms like viruses happened. And that is how, you know, the relationship of viruses and bacteria is to rely on other hosts to make them live. Okay, so that's how they have evolved.
Jayesh, you also have your hand up.
Ma'am, are other species also evolved by other animals? Are other species also evolved?
Yes, other species also evolved.
Like is T-Rex evolved by chicken?
Evolved by chicken, and before that I couldn't hear.
T-Rex.
Oh, I see.
T-Rex evolved by chicken.
I see. Well, T-Rex is a very nice example of evolution, because during the evolution, T-Rex have disappeared. So they have not evolved into chicken. Rather, they were so large for the ecosystem that they couldn't survive on their own. And survivability, what is the term to use, that what is the survivability of a species of dinosaurs, was less, because they couldn't survive in this new ecosystem. So they largely disappeared. There are stories of a lot of things, and we can go on talking about it. But yes, chicken, I'm sure, has other ancestors. Yes, go on. I think Advik has a hand since like a long time. I want to address his question as well. Advik?
Ma'am, I was talking about that the all drugs can also damage our cells, because they cannot identify who is who.
Yes, very true, very true. And that is why, Advik, before formulation of a drug, there are an army of scientists that try to understand that how much the drug has to be given, which is why, you know, we say that 1 tablespoon or 2 tablespoon, and in what condition it has to be given. And these experiments go on for a very long time on human bodies. Before human bodies, we do it on animals. So these are called clinical trials. There is a lot of research on the effect of these drugs on the human body.
मुझे ना एक चीज याद आई. I watched in a video that कुछ वायरसेस का हल निकालने के लिए उसके पहले ही उस पर रिसर्च करके उनका तोड़ निकाल दो.
Yeah.
चाइना एंड ग्रीनलैंड में होता है ना.
All right. Thanks for sharing that, Advik. In the interest of time, we can take just one last question. So both of the Arushas have their hand up. Either one of you can ask your question. Arush, yeah.
What if like we have less bacteria in our body and we take more antibiotics now?
Yeah.
The antibiotics start
Yeah.
destroying our
Ma'am, is there a scientist lab in Pune?
Yes, it's National Chemical Laboratory, also ICER Pune, all these places you can visit. You let me know if you have to visit.
Ma'am, can I visit the lab which you have told, which is in New Delhi?
Yes, we can arrange something. I just quickly want to answer the question of Arush that yes, Arush, antibiotic resistance is a very big problem when we take antibiotics mindlessly, meaning that if we do not ask the medical doctor before taking antibiotics, because he is able to judge and see and check whether the bacterial infection is severe or not, then only antibiotics should be prescribed. If the bacterial infection is not so much, then antibiotics can really harm because the bacteria tries to escape it. And that is called antibacterial resistance. And when that happens, then even if you take another antibiotic, it will not help you. So it's very important to take antibiotic only when the medical doctor is writing for it. Yeah. I think there is one last question by another Arush, but he's on mute. So
Arush, we can't hear you.
Ma'am, when we take the medicine, so the protein is breaked, so it replaces, right? So it is in the form of that shape, or it happens in the form of shape when it's gone into the stomach.
yeah. So Arush, firstly, let me tell you if there was confusion. Most of the drugs or most of the medicines are small molecule chemical compounds. They're not proteins. Proteins can also be the delivery mechanisms of as medicines, which is what I think what you're referring to. And you're right, it has to be stable and in particular shape to be effective. Okay, so not all drugs are proteins, but proteins can be drugs as well, especially the molecules that you are referring to, the ones that go to the stomach, and they have to be digested. So they all have to be in specific shapes. Also hormones and so many other molecules which are proteins. Yeah.
Right. All right. I think that